In this video, I am going to introduce the HPA axis, that is, the hypothalamus anterior pituitary adrenal cortex axis. In this particular video, I'm focusing only on the H and the P components. Here we can see the anterior pituitary gland, median eminence, and ultimately out into the hypothalamus. I'm going to start this story by talking about what stimulates this axis in the first place. Well, various parts of our brain, including the prefrontal cortex, right, this kind of higher order thought part of our brain, the hippocampus, the part that is responsible for forming memories, the brainstem, which regulates different kind of basic functions like heart rate and breathing, and other parts of the hypothalamus responsible for maintaining homeostasis. Each of these parts of the brain can sense a stressful situation. These types of stressful situations are not necessarily running away from a bear or needing to, immediately get yourself out of danger in this very second. Instead, these are more long-term stressful situations. So maybe you are starving, maybe you are, kind of kicked out of your tribal group, and therefore you don't have the benefit of the other members of your tribe to help you get food and shelter, and so this is a very stressful situation. Maybe even you are sick for a long time. Okay, so stressful response is detected in any of these places and ultimately can trigger the HPA axis, therefore the release of cortisol and the stress response via the paraventricular nucleus of the hypothalamus. These cells can release corticotropin-releasing hormone, a 44 amino acid long peptide, in response to messages from the rest of the brain. Now, I have greatly simplified the messages from the rest of the brain into two different neurons. Right? It's not just two different places that are stimulating. I have just very much simplified this. Here we can see glutamate, a neurotransmitter, is released by some of these neurons from the rest of the brain. This can trigger the release of corticotropin-releasing hormone into the first capillary bed of the portal system. Acetylcholine is another stimulatory neurotransmitter, and GABA can be released to inhibit corticotropin-releasing hormone. The sympathetic nervous system can stimulate a long-term stress response by norepinephrine binding to adrenergic receptors on the paraventricular nuclear cells. Okay. In addition to CRH being important in this process, or in the stimulation of the HPA axis, CRH can also feed back and have a direct impact on the rest of the brain. Here we can see there are CRH receptors in the amygdala, which means that the CRH itself can actually affect emotions, kind of these more aggressive behaviors sometimes, directly without even needing to stop and make cortisol. The substantia nigra is one of the basal ganglia, and this is responsible for essentially regulating appropriate behaviors, appropriate movements, and so CRH can directly affect behavior. Okay. The cerebral cortex, so CRH directly, in addition to cortisol, the stress hormone, can impact higher order thinking. It can affect memory formation and also the sense of smell. Okay, so CRH is very important in the stress response. As we're going to see, CRH is going to stimulate the HPA axis, but also a second hormone can enhance the response of CRH. And that hormone is called antidiuretic hormone, also called arginine vasopressin in humans. This is traditionally considered to be a posterior pituitary hormone because it was released from the posterior pituitary, but also we can see that some neurons release AVP into the portal system as well, which is then delivered to the anterior pituitary gland. Now, as we can see in the anterior pituitary gland, there are actually two different types of cells that are going to be important to this story. The corticotropes, or corticotrophs, are found in the pars distalis, and they are very abundant. The melanotropes are found in the pars intermedia. Now keep in mind that the pars intermedia is most prominent in fetal development of humans, but ultimately it's going to atrophy over the course of our lives. And so the function of melanotropes is going to shift elsewhere in the body as we get older. Let's zoom into corticotropes. This is a corticotrope. We can see that CRH released from the paraventricular nucleus binds to a G protein coupled receptor on the surface of the corticotropes, stimulating the second messenger system cyclic AMP. Cyclic AMP is going to increase the expression of a protein precursor molecule, a prohormone, for this hormone right here, adrenocorticotropic hormone. In response to CRH, adrenocorticotropic hormone is going to be expressed, and cyclic AMP induces an influx of calcium to trigger the exocytosis of adrenocorticotropic hormone. There are also some other hormones that are released simultaneously, which we'll talk about in just a second. The arginine vasopressin binds to its receptor, V1b, one variant of the vasopressin receptor, and this particular receptor induces an IP3 DAG second messenger system, which ultimately enhances the responsiveness of this G protein coupled receptor to CRH. So essentially, AVP enhances the release of adrenocorticotropic hormone. Okay. This prohormone is called POMC. It is the precursor to not only ACTH, but a variety of other hormones as well. POMC stands for pro-opiomelanocortin. The reason for that is that it is a prohormone that produces an endogenous opiate, a hormone that stimulates melanocytes, and a hormone that is going to induce the production of different steroid hormones. Okay, this POMC is a very long protein. At the amine terminus, we have 16,000 amino acids. ACTH is a 39 amino acid peptide, and this is essentially cleaved from these amino acids as well as these, which I'll talk about in just a second. And again, in response to CRH, it is going to be released into the blood. Okay. Now, there are other enzymes that can actually cleave some of these other hormone fragments here. They can free them from the larger POMC chain. The one that I want to mention now is beta endorphin. Beta endorphin is an endogenous opioid. It binds to opioid receptors that we might think of when we think of taking morphine or something. And essentially, these receptors are there to bind to molecules such as this from POMC released by our own body. Beta endorphin in particular acts mostly as a neuromodulator. It hyperpolarizes different cells, different neurons within the brain that are responsible for producing a behavioral response to pain. And so this opioid essentially suppresses the response, suppresses the severity of response to pain. Okay. Also, it increases appetite. Okay, it's anorexic. Okay. So both of these, ACTH and beta endorphin, and to an extent gamma LPH, we're not going to talk about that one, both of these are released into the blood of the portal system in the anterior pituitary gland. Now, in the melanotropes, different hormones can be released. So let's see what happens there. In a melanotrope, as a fetus, or in a lot of other animals, ACTH can be further broken down into two additional hormones. The first 13 amino acids of ACTH is alpha melanocyte stimulating hormone, a-MSH. When this hormone is released, it binds to receptors in melanocytes, which are cells in our skin, which are responsible for making melanin, which is the pigment in our skin, or one of the pigments in our skin, as well as in hair and fur. And so when a-MSH binds to the receptors in the melanocytes, there is an increase in melanin production. And so this is really important in fetal development, but of course the pars intermedia degrades over time. Okay? So in humans, that is, and so this function actually switches into the skin in adults. Okay, so there's actually ACTH, which enters into these skin cells, and an enzyme cleaves MSH from ACTH within our skin cells themselves. Okay. In other animals, not humans, the pars intermedia remains throughout the course of their lives, and so this melanin stimulation is really important to particularly animals that change colors, maybe to white in the winter and brown in the summer, both for camouflage. Okay. The MSH receptor also binds to ACTH, right? So this entire hormone here. And so that is important because if there is a prolonged or a particularly strong stress response, ACTH can also induce the production of melanin, that is the darkening of the skin, which of course is seen in some hyperstimulation of ACTH or a prolonged stress response. Okay. MSH, alpha MSH, is also important in modulating the immune system. It itself serves as an antimicrobial molecule and also can regulate the inflammatory response. Okay, so MSH is very important in a lot of different things, particularly in the coloration of the skin, hair, and fur. Okay. Now the rest of this molecule, the amino acids 18 to 39, are cleaved off as CLIP. Okay, so in summary here of the stimulation, various parts of the brain experiencing stress induce the release of CRH, a peptide hormone, into the first capillary bed of the portal system. AVP is also stimulated to be released, and it is under regulation of the renin-angiotensin-aldosterone system. Okay. CRH binds to its receptors on corticotropes, okay, which stimulates an increase in the expression of POMC. Specifically, ACTH is cleaved off and released into the blood, also beta endorphin. Okay. In melanotropes, ACTH is cleaved into alpha MSH and CLIP. Alpha MSH is released, as well as some of the other molecules from POMC. AVP can enhance this entire response by increasing the affinity of its receptor for CRH. And so that's all the positive stimulus. Negative feedback, briefly. Cortisol is the hormone that is ultimately produced by the adrenal gland of the HPA adrenal axis. Okay, so cortisol is going to suppress both the anterior pituitary as well as the hypothalamus. The anterior pituitary gland is suppressed when cortisol binds the glucocorticoid receptors within the cytoplasm of the cell. This interaction blocks the expression of POMC and therefore reduces ACTH that is released. I also want to point out that AVP can increase ACTH by suppressing the negative feedback. Okay, so again, cortisol blocks POMC expression. AVP increases the expression of POMC by enhancing this response and by suppressing the suppression. Okay, so that's how the anterior pituitary receives negative feedback. Cortisol can also act up here in the hypothalamus. Here we can see cortisol binds to its glucocorticoid receptor, which is going to decrease CRH secretion via binding to receptors within other parts of the brain. And so this neuron might secrete GABA, and so there'd be more inhibition of this neuron and therefore less exocytosis of CRH into the portal system. Okay. Another interesting way that cortisol can suppress CRH release is that it can induce CRH neurons to synthesize endocannabinoids. Okay, so two different types of molecules that bind to the same receptors as other cannabis substances that are exogenous, so not endogenous. These endocannabinoids can then have a retrograde effect on positive stimulation of these cells. That is, cortisol, if levels are high, can induce endocannabinoids to inhibit the release of glutamate. So there's the suppression of a positive stimulus from elsewhere in the brain due to the presence of cortisol. Now, one final note here. Remember that AVP can enhance this entire response. At no time did I mention how cortisol suppresses AVP action, and that is on purpose. AVP is not actually under negative feedback from cortisol, and so even if CRH is suppressed, AVP can enhance the responsiveness and therefore ultimately trigger the release of ACTH by enhancing this binding to any remaining CRH that's in the blood. And so AVP can continue to stimulate the stress response even if this axis is under negative feedback. Okay. So once again, this has been a very brief summary of the HPA axis, specifically the hypothalamus and anterior pituitary gland axis. We've looked at how CRH is released from the hypothalamus under stimulation from elsewhere in the brain, experiencing stress. CRH induces the release of acetylcorticotropic hormone from the corticotrope or corticotroph cells. That hormone is released into the second capillary bed of the portal system and is later going to bind to the adrenal cortex cells. Melanotropes and the pars intermedia are very important early on in life. Later, the alpha-MSH is no longer really produced in the melanotropes. Instead, it can be produced in the skin. Thank you very much for watching. I hope you enjoyed the introduction to the HPA axis.